Good afternoon. My name is Dwayne Brown with the Office of Communications, and welcome to NASA headquarters. Today you will hear about the Curiosity rover's analysis of the first sample of rock powder ever collected on Mars. We'll have brief presentations from our panel, then we'll open it up with questions starting here on the phone lines at centers, and also have our social media community have an opportunity to ask questions. Of course, you can get all of this information and the latest updates on Curiosity and all of NASA's Mars missions at www.nasa.gov/Mars, and join the conversation in the social media community. And there are a lot of people all around the world talking about Mars. Send your question into hashtag AskNASA and on Twitter @MarsCuriosity. Before we begin, coming to the podium to put things in context for everyone is the Associate Administrator for NASA's Science Mission Directorate. Please welcome Dr. John Grunsfeld.
Well, I'd like to welcome everyone to this press conference. This is just incredibly exciting for me. And I think once you hear the results, and I've not heard the detailed results yet, I think there'll be a tremendous amount of excitement. When we think about the Mars Science Laboratory event that we had last August, the landing of Curiosity on the planet Mars, often what comes to mind is the 7 minutes of terror. Well, since then we've had 7 months of anticipation for the results that you're going to hear about today. For me, one of the most remarkable things about the Curiosity rover is that everything is working. And I think that's just an incredible engineering feat. And I want to remind everyone who's listening that the amount of work that went into the results you'll hear is really been just incredibly intensive, has pushed human technology, has pushed what we're able to do not only on Earth with instrumentation, but on a planet that's tens of millions of miles away in a very difficult environment. I also want to remind everyone that what you're going to hear about are not the discoveries of Curiosity, but the discoveries of scientists here on Earth. And represented here today are those scientists. And it's not just the 4 folks who are up on the stage, but it represents hundreds of scientists working days and nights. And I think they've probably been doing ops, on nights and weekends, not because they have to, but because they love it. Because this is what excites them. The fact that we're on Mars and that we have this analytical laboratory on Mars that's being controlled by the scientists and engineers and technicians from here on planet Earth. So with that, I will hand it over back to Dwayne. And, I think this is going to be incredibly exciting. I know my anticipation is very high. Thank you.
Thank you, John, and that's a great segue because now I get to introduce these incredible scientists, a small part of a tremendous team. First up, you'll hear from Michael Meyer, lead scientist, Mars Exploration Program, NASA headquarters, Washington. John Gartzinger, Curiosity project scientist, California Institute of Technology, Pasadena, California. David Blake, principal investigator for Curiosity's chemistry and mineralogy investigation at NASA's Ames Research Center, Moffett Field, California. And Paul Mahaffey, principal investigator for Curiosity's sample analysis at Mars Investigation at NASA's Goddard Space Flight Center in Greenbelt, Maryland. So with that, turn it over to you, Michael.
Thanks, Dwayne. The NASA Mars Exploration Program has progressively approached the red planet from a global perspective to focus exploration of regions, past and present, that exhibit the potential for life. Every successive mission has boosted our expectations that Mars could have been a habitable planet, a place that could have supported life. This program of orbiters and landers have brought us to the point of seeking a habitable environment on Mars. This is what brought the rover Curiosity to Gale Crater. Mineralogical and geomorphological evidence from orbit, showing that the area had significant amount of water in its past. As John mentioned, on August 6th, Curiosity landed spectacularly where we wanted in Gale Crater. Within 2 months, the team found an ancient riverbed, evidence of flowing water, and we followed that downhill to Yellowknife Bay. At the same time, we exercised the rover's capabilities, tested the instruments for the first time, and doing science along the way. We have now completed all the first-time activities, including the first sample drilled on another planet. This mission has been a fantastic team effort of engineers and scientists to deliver a highly capable exploration rover to Mars. The rover is now fully commissioned for science. All the instruments are working, and the keys to the rover have been turned over to the science team. Woohoo! So Mars has written its autobiography in the rocks of Gale Crater, and we just started deciphering that story. So chapter 1, Yellowknife Bay. This was an ancient environment with the right elements, minerals indicating a near neutral environment, and slightly salty liquid water. All the prerequisites to support life, a habitable environment. And so for the rest of the story, I'll turn this over to John.
Great, thanks, Michael. It's a great science story, as Michael was saying, and I need to start first with acknowledging our colleagues that came before us, and also the entire planetary community that supported this mission. As you know, developing MSL was a tremendous challenge, and we had plenty of adventures there, but we got the support from our community, and we really appreciate that. I'd also like to thank the Mars community, and through the leadership of Matt Gollenbeck and John Grant, which led to the final selection of landing sites that ultimately led us to Gale, where we have had a terrific time so far. That's been great. And also in particular, the MER mission, Spirit and Opportunity, Mars Express, and also Mars Reconnaissance Orbiter. If we have looked farther, it's because we have stood on the shoulders of giants, and those missions allowed us to fine-tune our exploration campaign that led us to this place. Finally, those of us that get to sit up here today are joined by our colleagues back at JPL and elsewhere, the other PIs of the mission, including Ken Edgett, Ralph Gellert, Don Hassler, Mike Malin, Igor Mitrofanov, and Roger Weens, and Javier Gomez Elvira. Every one of the instruments has led into the discovery that we have made here. Some of those instruments presented back in January when we first talked about the geology and that ChemCam discovered the first evidence for sulfates in this area here. You'll hear more results coming out next week at LPSC. And then at EGU over in Europe in April, you'll get to hear more still. So this has been a very comprehensive exercise, and we didn't just stumble into this area. This is something that took a lot of planning. Okay, so let me go to the first display item and bring you back to where we were the night of landing when we as a community first looked at this slide. We had selected a landing site, and the landing ellipse in particular was close to Mount Sharp, which was considered to be our primary objective. And so you can think about drilling an oil well here. You don't just go in with one objective. You need primary objectives, and you want secondary objectives. And we had a secondary objective, which was the distal part of this alluvial fan that you see here in the landing ellipse. And we needed this in the back pocket in order to have the landing site confirmed by the review board and then eventually accepted by headquarters. And in case something happened to the rover, we needed to make sure we had science to do in that landing ellipse. But that was sort of you can think of it as a backup or a secondary objective, and it turns out now, in fact, to have become our primary objective at this point. We landed at there where it says Curiosity Landing Site, and we drove just a few hundred meters in the opposite direction. We did this deliberately, and this was based on the mapping that the science team did in advance of landing, and based on the previous mapping that came from Odyssey and MRO and all those great missions before us. And in this particular case, it led to the deliberate discovery. So it wasn't serendipity or luck that got us here. It was the result of planning. Now, what Paul and Dave will tell you about is the part that we do consider serendipitous. We had no idea that we were going to go into the aqueous environment that we were predicting to exist here, and also find sulfates and also find clays, and those guys will tell you about it. So that's one of the reasons that we're going to be spending some time here. So let me turn it over to Dave, and he can tell you about Kemin.
Well, thanks, John. And we got really excited when we first saw these bedrock at John Klein and saw these concretions. And the reason is concretions are evidence of a water-soaked sediment, a soft sediment. But what kind of an environment was it? Was it ever habitable for life? And if it was, would it preserve the organics for literally billions of years until we came here to take a look to see if we could see what was there? If you turn to the first graphic, you can see what made us think we really found something special. Okay, well, this is what we call pay dirt. This powder in the scoop here is from John Klein, the drill powder, and it's gray-green, meaning that it wasn't highly oxidized. And you can see in the back of the scoop there's a little bit of reddish material. This is from the rock nest, and this is highly oxidized. So anyway, what it shows you is that this material was never highly oxidized, and therefore, if there was organic material present there, it could have been preserved. The second graphic shows a comparison of the two X-ray diffraction patterns that Kemin has collected so far. On the left is rock nest soil, and on the right is the pattern we got recently from John Klein. You can see they look very similar, and from our analyses, we can tell you they both have igneous minerals, feldspar, pyroxene, olivine, and magnetite. What's different, if you look at the John Klein diffraction pattern, down close to the central point there, the intensity is due to clay minerals, and you see they're labeled phyllosilicates. And we can tell you from our analysis there's between 20 and 30% of a phyllosilicate called smectite, and that smectite forms in the presence of water. We know that. In addition, we have evidence of salts like halite and calcium sulfates rather than iron or magnesium sulfates that were found at Meridiani. And this suggests that the water was relatively neutral pH, and in other words, it was a potential habitable environment. So all of this is what mineralogy can tell you from an ancient surface that's billions of years old. So the next graphic shows you what we think a good terrestrial analog is for this material we found in Yellowknife Bay. The left image shows a clay-bearing sediment deposited in a lake bed in southern Australia. And on the right, you see a core of the sediment, and the different layers in the core represent different changes in mineral composition as the lake sediment was deposited. With that, I'll let Paul talk about what the SAM instrument found.
Thanks, Dave. Just delighted to show you some results from SAM. And I'm going to explain a little bit about how we did this fairly complex experiment. But I thought it would be fun to bring along what's a full-size scale model of SAM, the Sample Analysis at Mars experiment. SAM and Kemin are both buried deep inside of Curiosity, so in these kind of beautiful self-portraits that Ken Edgett's camera takes of Curiosity. You don't see much of SAM and Kemin. We have a test bed up at Goddard. You don't see much of it either because it's an environmental chamber. It's buried deep inside an environment that represents Mars. So here we've kind of taken away the aluminum paneling and put on plexiglass and made a model. And where the experiment starts that I'm going to describe, we have just a little bit of sample located inside a SAM cup. And I went last night into Amy McAdams' lab up at Goddard and dug around and found some nontronite, which is a clay mineral of the type that we're going to be talking about today. And there was a scale in there, and I weighed out 45 thousandths of a gram of that stuff because that's about the amount that was in our SAM cup when we analyzed it. So that's where the story of this analysis that I'm going to tell you starts. We have the sample in the cup in SAM. We have loaded it the previous sol, the previous day, and we're ready to do our analysis. So it's night on Mars. The rover's gone to sleep. Sam's kind of a night owl. We like to operate at night, and nobody else there to bother us. But it's also a good thermal environment for some of the instruments to operate. And so we had put the sample in the cup through this little inlet tube. This vibrates as the sample's going into the cup. And then the sample manipulation system developed by our collaborators at Honeybee Robotics can be seen down here. And it turns out that the way the sample gets to the oven is this little carousel rotates. The sample's dropped into the cup. The oven, the quartz cup moves over, and then it raises into the oven, a very small oven, where we take the sample up to the maximum temperature that I'll show you. And what we do then is we start heating up the oven. We get a flow of helium going over the sample. We heat up the oven, and then with the mass spectrometer, which is right in this area, we sniff a little bit of that gas, and we measure the chemical constituents that come off. And as we do that, we capture a little bit of gas in our tunable laser spectrometer that was developed by Chris Webster's team out at JPL. And we capture a little bit more of the gas in a hydrocarbon trap, because one of our objectives of this experiment really is to search for organic compounds on Mars. And we later will send this gas to the gas chromatograph. I think I have a button here that'll make one of the columns light up. And then the gas goes through those columns, and the individual constituents come out one by one, and then back into the mass spectrometer through a back door. And again, we analyze what Mars is made of. And so if you go to the first graphic, we'll show you some of the data. And this really is just picking out the 5 major gases that were evolved from the sample. And let's start with what's labeled water on top, but the mass that we're monitoring that is mass 18. That's the signature of water. And you see the temperature scale on the bottom going all the way up to 1500 degrees Fahrenheit in this case. And that water is coming off at really high temperature. And that's exactly characteristic of these smectite clays, and it's very good confirmation of what the ChemIn saw. We really do have clays here, and about 30% of the water that's coming off is at high temperature water. Go down to the lower left, you'll see a blue trace. That's oxygen. We've blown it up by about a factor of 10 in this case for illustration. And we did see some oxygen at our Rocknest dust pile, and we attributed that to the decomposition of a perchlorate, which is pretty interesting. It looks like there's very likely some perchlorate here as well. The red peak is likewise carbon dioxide. The carbon dioxide is produced either from oxygen reacting with carbon in the sample and making this carbon dioxide, or really the other alternative is a decomposition of a carbonate. And both of those possibilities are just fascinating, so that's what we'll be pursuing as we progress with new samples and so on. And then finally in the bottom right, at higher temperatures, you see masses labeled 64 and 34. And those represent an oxidized and a reduced form of sulfur. They represent respectively sulfur dioxide and hydrogen sulfide. And so that's just fascinating. We have both oxidized and much more than in this atmospheric dust, much more reduced sulfur there as well. What the tunable laser spectrometer was doing in the meantime in this experiment was measuring the deuterium to hydrogen ratio in water. And a very interesting observation, we had measured a very high deuterium to hydrogen ratio in water evolved from the dust. And we understand that as being a signature of a good fraction of water having been lost from the Mars atmosphere over geological time. And in this sample, we see just the lowest deuterium to hydrogen ratio that we've seen in evolved gas so far. And so that's something we're definitely going to be pursuing as we go forward with other samples. So go to the next slide. And here's what the search for organics is looking like. The data looks like signatures of mass to charge, just as I showed in the previous time. But here these compounds are coming out of the end of the gas chromatograph column. And here we see 2 compounds that we actually had also detected at Rocknest. Very simple chloromethane and dichloromethane compounds, and it looks like they're above the background level. It looks like they're there. We have to be very careful at this point in interpretation. This was the very first sample that had gone through the Curiosity drill, and so there's always a possibility that some residual carbon that was on the drill bit made its way into this sample. So we're really looking forward to repeating this experiment and seeing if these signatures of simple chloromethane compounds persist. So the really good news is the instrument is just working beautifully. It's a credit to the very talented team that worked hard on not only making this stuff, but making it robust and making it work in this very difficult environment on Mars. So with that, I'll turn it back to John for some additional comments.
Great. Thanks, Paul. So what I'd like to do now is sort of set the stage a little bit for what we view in this mission as the transition from the original goal a decade ago of the search for water on Mars to now the search for habitable environments on Mars. And if we go to the first display item there, what we can see are two rocks separated by a decade of research. The one on the left is from the Opportunity rover back in 2004, a rock called Watmay. And what you see here is a rock. These images have been processed by Mike Malin and Jim Bell with what's called white balance, and it helps bring out our terrestrial intuition to sort of get a sense of what these rocks would look like if they were on Earth. The one on the left is basically from the sequence of rocks at Meridiani Planum, a rock that is reasonably fine-grained. The particles were either formed in water or transported in water. It was then cemented in water, converted from sediment into rock. And then after that, it was fractured, and then some of the fractures were filled in with what looks like a relatively thin material in this particular rock. But you see all the bumps sticking out. Those are the famous blueberries. These things we know are concretions. Well, it turns out these things are turning up on Mars, and here on the right is our rock in the Yellowknife Bay area called Sheepbed Unit, we've named it. And again, you can see it approximately has the same color on the surface. It's laced with these features that look like concretions to us. And the big difference is that you can see in that rock that it has a white vein fill running through it. That's the thing that ChemCam first hit and told us that there were probably sulfates here. So texturally, you see rocks that were transported in water, formed in water, cemented in water, altered in water, and but that's what you get on the surface. And so what we need to do is scratch below the surface, and if you go to the next one, this is what a decade of engineering gives you. On the left, there's a rock that was one of the first rocks that we ever interacted with at Meridiani with the rock abrasion tool. And on the right, we have the drill hole from Curiosity. And the drill hole is about 1/3 of the size of the rat hole there on the left. But the big story is in the powder that's generated. And so as we learned at Meridiani, we have a rock that is composed significantly of hematite in addition to the sulfates, iron-bearing sulfates that indicate very acidic waters. On the right, we get to see the new Mars, the gray Mars, that one that suggests habitability that has these clays and other minerals present. So what then do we mean by habitability? The key thing here is an environment that a microbe could have lived in and maybe even prospered in. So there's three things that we want to point out today that Dave and Paul have shown you. And the first issue comes down to acidity. We don't see any of the evidence that we have here, the rock on the left, the one from Meridiani. It's totally different in the subsurface in this rock on the right. We have the clay minerals, which form a neutral pH. We don't see the iron sulfates, which indicate acid pH. Instead, we see calcium sulfate. This rock, quite frankly, looks like a typical thing that we would get on Earth. And it's a neutral pH environment. And I think everybody has a sense of what acidity means, but there are some microbes that exist at very, very low pHs. But wait, there's more. And the second point is water activity. This is how much available water there was for a microorganism to live in its environment. So with that, I'm going to pull out a prop here. It's a jar of honey. Everybody always wonders why it is the solution of water and sugar can last on the shelf forever and ever without spoiling. And the reason why is that even though there's a lot of water in this honey, there's not enough that's available for a microorganism. And if a microorganism winds up in here, all the water will be sucked out of the cell. It's this thing called osmosis. The organism won't be able to live. Turns out the rock on the left there, that's what we think happened at Meridiani, but instead of sugar, we had a salt called magnesium sulfate. And there was so much of it that it would have inhibited microorganisms to live there. That was not a habitable environment. And then there's one more thing that we're really excited about that we found at at Gale, and it's a battery. And basically, these minerals that Dave and Paul were telling you about, they're effectively like batteries. Some of them are negatively charged, and they have various oxidation states. And what we have learned in the last 20 years of modern microbiology is that very primitive organisms, they can derive energy just by feeding on rocks. So when Paul talks about sulfate versus sulfide, and Dave talks about clays and magnetite, these are the kind of things that tell you that there could have been a flow of electrons in the environment. Just like on this battery, you hook up the wires, and it goes to the light bulb, and the light bulb turns on. That's kind of what a microorganism would have done in this environment if life had ever evolved on Mars and if it was present here. So that's what we mean by habitability. You take all three of those factors, and to really understand that, that's what we built this payload for, and that's what we feel that we've succeeded at. And so with that, I'll turn it back to Dwayne.
Thank you, gentlemen. Okay, we're going to open it up for questions. Now, normally I would start here in Washington, but we're going to go out to the West Coast first and then come back to the Jet Propulsion Laboratory.
Hi, how's it going? NBC in Los Angeles. So can you talk to me a little bit about the area where the rock was found? What would it have been like in ancient times? What would we have seen there?
Okay, so what we imagine it would have looked like was the picture that Dave showed. We feel is a pretty good representation. It's conservative in the sense that it shows a lake bed that's dry. The lake bed was filled by sediment derived from streams, but we don't know how long lived it was, and so that's always a challenge we've got on Mars. It's not like the rocks come with numbers on them to tell you how long the water was there or how much there was there ultimately. But we believe that we wound up in this sheepbed unit at a place that was wet for a relatively long period of time, enough for all these chemical reactions to occur.
Okay, we're gonna come back to NASA headquarters. Any questions from our media here in the front row? Okay, so what I will do then is let's go to our social media community for a couple of questions that have come in to me. And then we're going to go to Alan Boyle, but let me take the social media question first. Clearly, NASA knows how to build rovers. What will the 2020 rover look like, or be able to do the same capabilities?
One of the things that we're planning with the 2020 rover, we now have a great capability in the entry, descent, and landing system and the rover system that we built for Curiosity. We want to use as much of that as possible in the 2020 rover, and so the approach that we're taking is to use that descent, landing capability and the rover as a platform for the 2020 mission, but we're going to populate it with new instruments and new capabilities, maybe in the arm, and for instance maybe doing coring instead of drilling. So that's sort of in the mix, but we're going to start with the basic rover and a system to get it to the surface safely. And because of that, I think we will be able to let's say, not have to do a lot of the engineering development that was required for Curiosity, save a fair amount of money, and get also a very capable rover system to the surface of Mars with the newest and best instruments that we have available at the time.
Okay, now let's go to the phone lines, and I believe we have Alan Boyle up first in the queue. Alan?
Hi, thank you. Alan Boyle from NBC News. I just had a general question about how do you feel that it sounds like you're all excited about this, and do you feel like it's mission accomplished? What went into nailing down these sorts of results? Can you describe a little bit of the emotion behind the process? Thank you.
Well, I'll start with sort of the whole idea of where to land on Mars with this tremendous asset. When it came down to it, we narrowed our landing sites down to 4, and all of them looked really good. And as you might imagine, every single scientist had their favorite. And so we had 4 major contenders. And I kept on thinking, now that we picked Gale Crater to go to, that the odds are 75% that we picked the wrong place to go. Well, you can imagine relief landing there, and then almost right off the bat, we do find evidence of water. We see an ancient riverbed. We're now finding an environment in the near subsurface, not too far beneath the oxidized layer, of finding sort of a neutral rock. All the things that we were really hoping for to find a place that could have been habitable in its past. So as far as I'm concerned, this is fantastic. All the rest is gravy in terms of how the rover is going to go about looking around this area, because it definitely was all the indications of being a habitable environment at one point in time.
I think I'll add to that by adding an additional point that what this does now is that I think with all the planning that Michael was talking about, going into picking the landing site and then effectively exploiting the landing site, we had 400 team members, and before we landed for about 2 months, folks got stuck into mapping this, and that helped us expeditiously find a place that when we landed, no matter where we landed, we would be able to move there and do the instrument checkouts and hopefully a good place. I think what we can do now with the issue of habitability in the bag, with a really good habitability now, we can undertake a more systematic search for a brighter carbon signal, the kind of thing that Paul was talking about. The search for organic carbon is an issue of this mission. And you want to do this as deliberately as possible. You don't want to just wander around and try stuff out. And so we have a search engine now, and there's 3 components to this, as I've stressed in the past. The one is that the primary environment has to have a mechanism by which you enrich whatever carbon signal is there, whatever reason it's there. You'd like to have a good, strong signal. But the problem is that what you see going on in these images is that there's a process that geologists call diagenesis, and it's all this chemical transformation that happens. Sometimes when you make these minerals and chemicals, they actually use the organics as part of this electron flow I was talking about. So they can be degraded. And then you leave the rock sitting around for a couple of billion years, and it's getting bombarded with radiation. This is not a simple problem, and it's going to take us a while, but I think the mission is up to it, and we're really excited to get started on that now. Maybe Dave and Paul want to say something.
Well, I don't know. I think we really ran the table with this analysis, because just coming to this place and analyzing this bedrock, finding an aqueous environment, finding what appeared to be a habitable environment, and with good preservation potential, it kind of covered all the points of what I would call just an elegant sample to analyze with our capability. So I'm really happy we landed at this spot.
Yeah, just add, this is really a different new set of tools with ChemCam on the mast and the survey tools we have, the first definitive mineralogy for a range of minerals with ChemIn, and the capabilities we've described with SAM. And the team is just delighted to be waking up every morning and looking at what's happening on this different planet. It's just tremendously exciting.
The next question is from Dan Vergano of USA Today. Dan?
Hi, thanks. Dan Vergano, USA Today. I guess this is for John Grotzinger, or really anyone who can handle it. In the past, you seemed a little pessimistic about getting this kind of signal from this site on the first drilling. What does it mean to have sort of pulled out these chemicals on this attempt about the whole environment of Gale Crater or Mars?
Well, I think that if the pessimism you refer to is that we go into this type of exploration with our guard up, I think it's justified, because when you land on Mars, strange things can happen, and I think if there's a positive message here is that we're getting better at this, and I don't think it's by accident. I think it's the result of the agency having a program of missions that build on top of each other, and that we can now be more predictive about what we're doing, but you still never quite know what's going to happen till we get there. It just so happened that the environment that we've been exploring is one that we have a lot of experience with here on Earth, and I believe that our science team was well prepared for that to really focus and figure out where exactly to drive the rover. But you always need to be careful when you're working on Mars.
Next up, Reuters, Irene.
Thanks, Dwayne. Congratulations. This is pretty exciting stuff you guys are reporting today. I had two questions. The first is what else needs to be done for analysis of the organics? You mentioned a little bit about that the assessments were preliminary, and then the second question probably is for John. I know this is not a life detection mission, but given that you've scored a hole in one so early, how much farther can you push this through the remaining 18 months of the primary mission?
Well, let me start with the organics. We're planning, when we had a pause to look at these software issues, had planned to do 2 more samples. One where we really, instead of one dump about the size that I showed, we put in 3 dumps and really try and increase the signal from very trace compounds. But what we're really looking forward to is getting the second drilled sample where we've really flushed out the drilling system, accomplished 2 things. Number one, there may be a little bit of residue from rock nests still in there. You saw it, in fact, the picture that Dave showed. There's a little bit of that coating on the sample transfer chain. And so we'd like to dilute that out further. And then very long term, we'll have to decide exactly when we decide to use what's our wet chemistry cups. And we have 9 of those, and that'll be a very deliberate decision, and we'll not do that until we're ready. But at some point, we're really looking forward to doing an entirely different type of chemical analysis.
I guess, Irene, the answer to the second half of the question is to underscore what you said, which is that we're not a life detection mission. If there was microbial metabolism going on, we really wouldn't have the ability to measure that. And if there were ancient microfossils in the rock, as good as Molly is, I mean, it can tell us definitively that we have a mudstone here, but it would not be able to resolve individual fossil microbes. What we can do is to survey additional targets that we have picked out, and we still want to go to Mount Sharp, and we hope to get there. And there are different combinations of minerals that we see from orbit that give us different prospects, and what I hope will become a burgeoning new field of comparative planetary habitability. And what that means is that if you look at how we've studied the ancient Earth, and you look at the minerals and compounds and substances that are available, and you look at the ways that different prokaryotic microorganisms can do their metabolism, they use different materials. It's almost like an organism has evolved to exploit every one of these little rock batteries that exists in the record. And so the question is, how many of these different kinds of batteries can we find at Gale Crater? And I think that really becomes our mission, along with the search for organic compounds.
Irene, did you have a follow-up?
Give us a little update on the status of the rover now with the recovery from the computer reboot and the shutdown for the solar activity.
Yeah, I mean, things are looking well. The rover is still on side B, and it's operating just fine. They're still suspending most of the scientific activities for a couple more sols as they sort through what they think was the root cause in the problem of side A, and because they want to make sure that that is a confirmed operational backup for any more operations. And as you mentioned, solar conjunction, we're headed toward that. Basically, we can't talk to the rover, and the rover talk to us for most of the month of April. And so what's going to happen is we'll do some more science activities now through the end of this month, permitting with the engineers confirming that things are safe for us to do those operations. But we will not do another drilling, the second drill hole, until after solar conjunction. So we're not going to start that activity until May.
Our next caller is from the Wall Street Journal, Robert Lee Hutz. Robert Lee?
Yes, thank you. Can you hear me all?
Yes.
Good. So, gentlemen, in a simple, straightforward, declarative sentence, please tell my readers what you have found here and why it is significant.
I'll take a swipe at that. I think we have found a habitable environment that is so benign and supportive of life that probably if this water was around and you had been on the planet, you would have been able to drink it.
I think that did it for him. Before we go on the phone lines again, let me take another question from Twitter. The Opportunity Rover, 3 months, and it's going on for many, many years. How long do you think Curiosity could last?
The half-life of its power is on the order of 84 years. So I expect the rover to be there to shake the first astronaut's hand if the astronaut goes to Gale Crater.
Next up, New York Times, Kenneth Chang. Ken?
Thanks for taking my question. I was wondering, given that these rocks had good preservation, was there a hope or expectation that they actually would have a stronger organic signal? And what does it mean that you don't have a stronger signal?
Go ahead, Paul.
Yeah, I mean, I think we try, since Mars is such a different planet, to go in as much as we can. At least I certainly do, with an open mind. It's one reason we have kind of the broad chemical survey capability we have on SAM. We're looking for signatures wherever they might be. And, I suppose, on Earth it's, finding organics in very, very ancient rocks is a difficult proposition. And no great expectations. We're just looking for the environments that are right in the first place and looking at it as best we can with the tools that we have and see what we see.
Ken, if I can, I'll just add a little bit to that. Paul's reference to the early Earth is, in our history of exploration there, you have to have a search paradigm. And that paradigm gets built on your understanding of the processes that result in the preservation of organics in the rock record. And one of the big things on Earth is that because of plate tectonics, we have a lot of heat that of course exists today. So a lot of organic compounds are degraded in the presence of that heat. On Mars, we actually think the planet cools with time, and so it may not be that heat's the problem, it might be that radiation is the problem, something that we're not so affected by on Earth. So we have these three factors, as I said before, to reiterate them. I think we're all going to have to learn these. The first is the primary concentration mechanism. The second is that all that cool chemistry that creates the habitable environment, including the presence of water itself, is not necessarily a good thing for the preservation of organics. And then the third thing is the radiation environment. And so our trick is to find a place where all three of those things went right, and that could take the entire length of this mission, but we're going to give it our best.
Okay, I just want to remind folks that, particularly the folks who are viewing this program, the community, the social media community, join the conversation at Mars Curiosity and send in a question, #AskNASA. Our next question will be from Nature, Alex Whitsey. Alex?
Thanks. I have a question for John Grotzinger. What does this mean for getting to Mount Sharp? You talked about primary objective becoming now to explore the area. When might you begin a traverse to Mount Sharp?
Well, like Michael was saying, we do, whether we like it or not, we have solar conjunction coming up here. But we've discussed this as a team, and as soon as we get past conjunction, we're gonna jump right into drilling a second sample, because as you know, it's very important to confirm what you've gotten before. With Rocknest, we analyze a couple of scoops, and our goal here is to do a couple of drill holes. And depending on what we find, because we're discovery driven, if it continues to look promising, we'll do some more work. If not, we'll get on the road and we'll probably clean up business with a few things that we pass by to help characterize the overall broad environment. But our decision as a team is to finish up here at John Klein and then hit the road to Mount Sharp.
Okay, I'm probably going to get in trouble for this, but I'm going to do it anyway. I'm going to turn the mic over to the head of the planetary division, Dr. Jim Green.
Thank you, Dwayne. The one question I have then, based on the observations and what you found out today, is would you say that Mars was habitable before or about the same time as Earth was in the history of the solar system?
That's a good question, Jim. I'm not sure we'll ever really be able to address that with our payload, but we've got a couple of different options here for the age of just relative to Mars, how old these things are, and quite frankly, they go between being as young as that alluvial fan lobe that comes down, which I think would be relatively young in the history of Mars. And it could be quite old. Maybe these rocks are somehow related to the base of Mount Sharp. We can't rule out that we're not looking at the base of Mount Sharp right now, in a way. So we got a lot of options open before us, but I think in any one of those versions, we're talking about older than 3 billion years ago. And we're probably looking at a situation where plus or minus a couple 100 million years, it's about the time that we start seeing the first record of life preserved on Earth. It's a great comparative planetary question.
Make a good reporter, Jim. Make a good reporter. Okay, let's go back to the phone lines. Mark Kaufman from the Washington Post. Mark? Do we have Mark on the phone? Okay, let's move to Craig Covalt. Craig.
Hi, this is Craig Covalt with SpaceRef Curious Mars. A question on the clays. If one of the more significant findings is that you had abundant water flow through the clay, how does that relate to meteorite findings where they have also identified significant water clays found in Martian meteorites? Then I have a follow-up.
I didn't quite catch
clays, white water, and then how does that relate to Martian meteorites? Particularly.
Well, I think the clays in Martian meteorites were just almost trace quantities, and the Martian meteorites that we've seen are mostly purely igneous rocks. The clays in this rock, which is a mudstone, which was something deposited in a shallow aqueous environment, are really a major percentage of the rock, and so they really represent a significant process. And plus, I guess you could call meteorites the ultimate what we call float rock. It came from someplace else, and we don't know where it came from. We know where this stuff came from. It came from this bedrock in Yellowknife Bay, and so we know that this environment existed in Yellowknife Bay with plenty of water.
Do you have a follow-up, Craig?
Question is, while I'm happy, there has been some chatter that you made a leak in one of your chemistry cells, correct?
Yeah, in the Rocknest sample, we certainly detected some vapor that was very easily identifiable. A trace amount of vapor, nanomoles. That's the signature of what's inside our wet chemistry cells. And so one way or another, whether it was through a very small leak through one of the pinch-offs or processing, a little bit of that vapor is there. what we are doing is trying to get smarter about how we do experiments to avoid some ambiguity with regard to where carbon coming from our sample comes from. And so what we did with these rock nest samples was we preheated the sample and let helium flow over it for something like 20 minutes. And it's a very clear signature of all that vapor being flushed out of the system. So that's really a very robust way, we believe, of getting around this issue that we saw at Rocknest. Yeah.
Next caller, Lisa Grossman from New Scientist. Lisa?
Hi, can you hear me?
Yes.
Great. I was wondering if you could compare this to Enceladus or Europa or other comparative potentially habitable environments. Is this the most certain habitable environment outside of Earth that we've seen yet, or the first that we are definitely sure is habitable? Can you put that in other context?
You guys take it. Yeah, I'd go for it.
I think the answer is yes. I mean, I think there was a question earlier about what's the take-home message about this, and I think this is probably the only definitively habitable environment that we've described and recorded. I don't know, there are places we would suggest could be habitable, but we haven't measured there.
Yeah, and again, here we're poking at what happened a very, very long time ago. It's not a current puddle on Mars. Mars is now cold and wet, and a lot of the atmosphere has escaped, and some of the radiation may have processed samples that we're looking at. It's in some respects, it's a fairly harsh environment. And so the focus of the mission is a lot on what things looked like in the past. That's kind of the idea both of this mission, where you can get down and drill into the rocks and do the types of experiments we've described, but also kind of the intent of an upcoming mission, which is going to launch this year, MAVEN, which is trying to study how the current atmosphere is escaping, and then through modeling try and get back to what early Mars looked like. So all these things are part of a program, and they all tie together, but it sure is fun to dig in the dirt and try and understand the ancient environment.
I guess one way to look at this is with Curiosity, we've been able to take this to the next level of understanding whether or not Mars is habitable. It's not just a broad brush, oh yeah, there's water and yeah, there's rocks. We actually know there's particular environments that water was there for a significant amount of time, that it was neutral, it wasn't too salty, there's plenty of water to not act like honey, but in fact water for microorganisms to use if they were there. And the idea that we have oxidized and neutral species in this place. So as we look into the details, it looks better. And for that reason, I would actually agree with Dave and Paul.
Okay, we're coming up top of the hour. We're going to be wrapping up shortly. I want to come back here to headquarters to see if there are any questions in the audience. I'll take a couple of more calls on the phone. Any questions here? Can you wait for the mic, please, and give your name and affiliation? Sorry.
It's Kitty Feldy with KPCC Southern California Public Radio. I know discovery is the exciting part. You don't know what you're going to find. But what could you, with the equipment that we have and the range that you're going to have, I mean, what are the kinds of things we could detect that could even make it more specific? I mean, what could we possibly find that is much more provable than what you found and told us about today as far as habitable conditions, or finding some more clues about life on Mars?
Yeah. Let me use that as a chance to go into the issue about the deliberate search for organic compounds again. As I said earlier, we need this paradigm, and it has to be a Mars paradigm. We can start first with our understanding of Earth, but as I already said, there's the three parts. The thing that to me, I think many of us on the team, that seems the most familiar for this environment, which is an alluvial fan and possibly a lake that was there, that's very familiar. And we can think about mechanisms that would have generated carbon, even carbon that comes from outer space, that would have accumulated there. After that, it starts to get different. The one at the very end, on Earth we have plate tectonics that tends to heat rocks up and destroy organic material. On Mars, we don't have that, but we do have radiation. The one in the middle is going to be, I think, the really fun one for the team, this process called diagenesis, because On Earth, we're used to a sort of a stable of compounds that we know about that can cause oxidation of organic compounds. On Mars, we've got a new cast of characters, some of which now are becoming familiar, things like perchlorates. There could be other things there as well that Paul and Dave will eventually tease out with their instruments that might be present there at trace quantities. And the question is, when you put those things in water and you add organics, how do they react in a way that might break them down? How fast does it occur? How aggressive is the reaction? And I think that's what I mean by comparative planetary habitability. We can begin to start to play that game now.
Okay, we're going to take one more question from the phone line, and then I'm going to invite Dr. Grunsfeld to come up for some parting words, and then I will conclude the briefing. And I believe we have Sky and Telescope. Camille, are you on the phone line?
Yes, hi. I'd be a bit of a long shot, but in December there was discussion might actually be some dust mound and not created. As far as goes, this sign of lake, does it have any effect on
Camille,
how Mount Sharp?
Camille, could you please repeat the question? You're kind of going in and out on here. Let's try that again. Can you repeat the question?
Sure. Let me try my hand. Is that better?
Yes.
Okay. At the AGU in December, there was discussion that Mount Sharp might be this sort of mound of dust, and it might not be from water eroding. As far as the scale goes, this former lake bed, is it a large enough scale that we can say how Mount Sharp was built, or is this pretty focused local?
I'll let the others chime in here, but we're actually not quite sure how Mount Sharp was built, and so the real evidence of how that happened is in Mount Sharp. And so what we're seeing now is what's at the base and what may have not necessarily pre-existed, but happened at the same time or even later. Looking here, we are at Yellowknife Bay, but how Mount Sharp was formed. We have to go to Mount Sharp to see those layers to sort that out.
Yeah, I agree with Michael completely. And just give you a heads up that when we start driving to Mount Sharp and you see us dragging our feet as we go along there and stopping to look at a few things, that's because we'll be trying to figure out how the rocks we're at now at Yellowknife Bay relate to Mount Sharp. That's how we get the relative age of all this stuff.
Yeah, good point.
Okay, so before Dr. Grunsfeld comes up, I am going to take one question going back to the West Coast to the Jet Propulsion Laboratory. JPL?
Yeah, this is NBC LA. John, this question is for you. Back in November, you said that there was going to be, the news was going to be one for the history books. Did you know now what you know now?
We got chapter 1.
Well, I can tell you I feel better about it now. You do, when that kind of a thing, you do need to sort of ante up. I think, basically, again, that expression at that time was just simply looking at the second run coming out of SAM and realizing that the instrument did a replicate of what it did the first time, and that if you ever got it into the right place, this would all pay off really well. And so it's just having confidence in the science team to get the mapping and provide the direction of where to go. And it seems like that's what's happened, and I certainly feel really great about it.
Okay, my director's saying we still have a few more calls, so if you guys don't mind, we may need to go over just for a few minutes. So there's a lot of excitement and interesting questions from folks, not only on the media calls, but in the social media front. So we're going to go to Alicia Chang of the Associated Press. Alicia?
Hi, thanks for taking my question. For John Grotzinger, I'm wondering how you can declare this spot a habitable environment without a strong organic signal.
Thanks, Alicia. That also gives me a chance to elaborate on something that I mentioned before, but I think it's often and easily confused. you don't have to have organic carbon present in a geological environment that's habitable in order to have microbial metabolism to occur. For example, almost all prokaryotic organisms, they don't use organics to metabolize. They actually use inorganic compounds. There does need to be a source of carbon there somewhere, but if it's just CO2, you can have, it's a complicated word here, but a chemolithoautotrophic organism that literally lives on rocks. It feeds on rocks. Add that CO2 there, and they will metabolize and generate organic compounds based on that carbon. So the fact that Paul is able to show in the SAM that CO2 spike, that's what we're real excited about there, because then that gives us our sort of desiderata of the key elements that are the building blocks for life: hydrogen, oxygen, phosphorus, sulfur, and then of course nitrogen, and then also carbon. And it can be inorganic to be utilized by a microbe.
And let me just make an announcement. We're going to take a few more questions, and these gentlemen will be available following the briefing for the media who want to have one-on-ones. So just call my office or the JPL office, and we will get these folks to answer some of your questions. The media that I will call on, please limit to one question so I can hopefully get us out in the next 5 minutes. So next up is Ken Kramer, Spaceflight Magazine.
Hi, thanks for taking my question, and really great results. I'd like to follow up on something John just said. I'm interested to know, maybe even from Paul Mahaffey, about the SAM run. Did you see any nitrogen or phosphorus or chlorobutanes, and can you talk about what was the weight % level of the phyllosilicates? Thanks.
So I think the weight % of the phyllosilicates is best answered by Kemin, and you gave a number for that of
20 to 30 weight %.
And did we see nitrogen compounds? We certainly are in our trace gases looking for those. It seems that we have some nitrogen oxides coming up. We're looking at compounds like HCN and so on. Deconvoluting those spectra requires work, and so we're working away on that. But it sure looks like we have nitrogen compounds there. In terms of phosphorus, we haven't seen any, but that really is an APXS result. They see phosphorus in their compounds. Very interestingly, we see a fair bit of hydrogen chloride coming up into our lines, and so the chlorine is not just producing these trace chloromethane compounds, but is also producing hydrochloric acid, HCl.
Next up, Peter Spots, Christian Science Monitor. Peter?
Well, thank you very much for extending this. The one question I have for John, I think it is, earlier in this series of briefings since the landing, you mentioned that there were some tests you would have to perform once you did find organics to see if they were homegrown or came from elsewhere. I wonder if you could recapitulate that a bit.
I'll just answer quickly and then see if Paul wants to add further to that. But as Paul outlined, we're not done yet here. We want to do some more runs to enhance the signal that he's seeing in SAM. But after that, if we got a positive detection that was reinforced and we know that if there's an organic compound that's not coming from the instrument and it's not coming from the rover, then we can feel confident that it does come from Mars. Then we got two options there. The one is that there's this sort of never-ending rain of cosmic debris that occurs on the surface of the planet, and so it could be that we're detecting that. And then it could be that it is actually being manufactured on Mars, and then you have to bifurcate again in order to show that the organic that you're seeing wasn't manufactured by some inorganic process, for example, in a hydrothermal environment. And then if you can discount that, then maybe you've got something that could be a trace of a biosignature. But that's a long row for us to hoe there.
Our last question is going to be from Mark Kaufman from the Washington Post. Greetings, Mark.
Okay, thank you very much.
Thank you very much. A simple question. Everyone has mentioned, has talked about Yellowknife Bay as being aqueous and references to possibly a lake. Could anyone be more specific? I mean, would it be accurate to say that at this point you believe that this was a lake or something like a lake? And if so, how large might it have been?
I think, Mark, you'll continue to see us hum and haw about that one. There's obviously this unit called the sheep bed, and the geologists can map this around and see that it extends around Yellowknife Bay, and that it projects under what we call the Gillespie unit, and so it extends underneath the outcrop to some unknown distance. So, just looking around the bay, that's sort of a minimum. And but the real question is, if this was a lake, how long, how deep, what was it like, the hydrology, and that's a much harder thing for us to reconstruct. So I think it's safest at this point to just say that we see something that's fine-grained. It could have been the material that was eroded from the crater rim that accumulated within some type of occasionally wet pool. But we don't have to go much further than that, and we'll just have to see. We can still collect more data.
Okay, now I'd like to invite Dr. Grunsfeld up for some remarks, and then I will close out. Dr. Grunsfeld?
Well, gentlemen, thanks very much for that description. First of all, congratulations. I mean, really incredible, and I know it's been years of hard work to get this far. I used to hike around as a young boy with a rock hammer and a collection bag here on Earth, trying to find interesting samples, and I have boxes of rocks. I was always on the quest for fossils, very hard to find on Earth. Even if you know where to look for them. But this has just been an incredible adventure just to get to this point and so early in the mission. That you must all be very happy. Someone asked, well, how do you feel? Just sitting in the audience here, I feel giddy. Because I have an image now of, possibly a lake, freshwater lake, on a Mars with probably a thicker atmosphere, maybe a snow-capped Mount Sharp. I mean, who knows? And so now we have the food to imagine a very different Mars than the one that you see behind us now, the modern Mars, the Martingale crater, which is a very dry, foreboding place. Having said that, though, and looking at the image and thinking about the work that you're doing with Curiosity on Mars, it makes me want to go. And so let me just ask you, how many of you now would like to go to Mars and be able to go with a rock hammer and maybe a little more modern instrumentation? Well, at least one hand goes up.
Just make the trip short between here and there.
I'll go, John, as long as you can get me back.
Yeah, well, I think that's a critical element, absolutely. And in fact, we are going, the fact is that we are on Mars. We're exploring Mars with Curiosity. We still have Opportunity roving, Mars Reconnaissance Orbiter, Odyssey, collaboration with ESA on Mars Express. We have Maven on the way, hopefully this year. That's the plan, to orbit Mars. We have InSight planning to land in mid-decade. We are participating with ESA on their ExoMars program, and then as Michael Meyer said, we're working on a Mars 2020 rover, kind of the cousin of Curiosity. And I think it's the questions that you're answering today. And this is a pretty big one. Did Mars? This is a huge science question, a planetary science question. Did Mars ever have a habitable environment? And we now have an answer. I think that's incredible, and we'll be able to follow that up with what we do on a rover later in this decade. In 2020. I think that's pretty exciting. So with that, I just want to give you my thanks for all the hard work and keep it up. I'm sure there's a lot more surprises that Mars has for us as we rove around. Thank you.
Okay, ladies and gentlemen, that will do it for us. I want to thank you for staying with us a little longer. Congratulations to the team. And again, keep the conversation going. It is going on Twitter and social media, www.nasa.gov/Mars for the latest results. And if you need to talk, the media that did not get a chance to ask questions or would like interviews, please contact my office headquarters, JPL, Ames Public Affairs, and Goddard Public Affairs. It's a big family here with incredible results. So thank you again. Thanks for joining us. Have a great day.